Welcome back, everyone. So we will start our third and last session in this symposium about cognitive evolution and environment with David. We recently moved from Arizona to Berlin, and we talk about mimicry and cognition. Thank you, David.
All right, thank you for that introduction. I'm going to be talking today about how signal categorization by foraging animals can depend on ecological diversity. Um, I'm really interested in why and how signals evolve. And we know from really basic models that in order for the evolution of signals to be stable, they have to be to the advantage of both the signal sender and the signal receiver. So, for example, this chemically defended nudibranch has a warning signal that this fish can receive, and it's the advantage of the nudibranch not to be eaten, of course, and it's also to the advantage of the fish not to eat the nudibranch because of the chemical defense. Um, but we also know that in addition to this basic mutual benefit to communication, that receiver psychology can also be really important to the evolution of signal form. So that's both how receivers perceive signals and how they learn about what constitutes a signal in the first place. So I'm going to be talking to you today about receiver categorization behavior as a driver of signal evolution. So to illustrate what I mean by this, we have a bird here, and it's trying to decide whether to categorize this butterfly as good to eat or bad to eat. And there are a bunch of different phenotypic characteristics that this bird could use to make its decision. Um, and this could actually be a really daunting task in natural communities, which can often be quite complex. So I'm going to refine my question a little bit and ask, how does the diversity of communities affect categorization behavior? I'm going to be talking to you today about a particular phenomenon called Batesian mimicry, which is really good for asking this kind of question. So in Batesian mimicry, we have a species that has a warning signal and an honest chemical defense, like this viper. And there's also a mimic species, which is undefended but has the same signal. And of course, this deceptive resemblance is confusing for would-be predators. So oftentimes, mimicry can be imperfect. So I've got examples of models over here on the left that are variously resembled by their mimics over here on the right. And imperfect mimicry is a really good way of testing hypotheses about adaptation, because mimicry is one of the rare instances in adaptation where we can form a reasonable null hypothesis about what an optimal adaptation looks like. Of course, adaptation will rarely ever be optimal, but in this case, we can ask questions about what is constraining these mimics from resembling their models more closely. And so we're going to be looking at how communities affect the evolution of mimetic precision. So one thing that the mimicry literature has put forth as a way that community structure could be important for this is the frequency of alternative prey in communities. So by alternative prey, I mean prey that are unlikely to be confused with members of a mimicry complex. So that would be all these gray butterflies over here. Now, in a community where there are few alternative prey available, predators might have to make a living by discriminating models from mimics and make selection for mimicry stronger. But if they're in a community where there are a bunch of alternative prey available, they might not be very willing to invest in learning to discriminate models from mimics at all. But this is only 3 species. Real communities are, of course, much more complicated. So I'm going to be talking to you today about different aspects of diversity that are measured in real communities. So the 2 simplest measures, actually. So the first one I'm going to talk about is species richness, just how many species occur in a community. And one hypothesis that's been brought forth about how predators might categorize prey as richness changes is that they might use key traits to categorize prey when communities become more and more speciose, or they have higher and higher richness. So this hypothesis was put forth by Beatty et al. In 2004, and they did a couple of experiments I'm going to explain to you. So in one experiment, they had a really simple community with 2 kinds of prey. and the prey were discriminable on the basis of both color and pattern. So there's this color is obvious, and then this pattern here is another trait that they could use to tell these apart. And they also had a rich community where they had a bunch of different colors, but the pattern was available to be used as a key trait. So it had the same number of states as the original community did. And what they found is that when they introduced these imperfect mimics over here, imperfect mimics were not very well protected relative to other kinds of prey that they introduced in the simple community, but in the rich community, the key trait was used in order to tell prey apart, and so these imperfect mimics were protected. But you'll notice that all of the colors that were used in the good and the bad prey were the same in these in this high richness community. And so the key trait is the only way that predators could tell apart these good and bad prey. So it's unsurprising that they actually use this. So to test this hypothesis more robustly, we'd want to design communities that have the same amount of information in the key trait and the other trait as richness changes. So I designed some communities that do exactly this. So in these communities, there are good prey on the top and bad prey on the bottom. And shape is always associated with being good or being bad. So all these circles are good, all these stars are bad, and then that's true for all these different shapes as we increase their number. The key trait is color here. So color is equally informative across all these communities and always has the same number of values, so either blue or yellow. So as community richness increases, predators are going to have to choose between using this 100% reliable trait of shape or using a less reliable trait in color. So this key trait hypothesis predicts that as richness increases, predators should shift from using the reliable trait above the x-axis here to using the key trait below the x-axis. Now, alternatively, the cognitive psychology literature has documented something called the relative validity effect. And the relative validity effect says that subjects should always use the more reliable trait to make a discrimination. Now, in defense of cognitive psychologists, they haven't actually explored a whole wide range of richnesses, but based on their data, there should actually be no variation in the use of a reliable trait. So human subjects make excellent generalist predators, and you can gather a lot of data quickly. So we made a game that is amenable to use by human subjects. Also, this is Andrea. She is my undergraduate and will be applying for graduate school soon, so keep an eye out for her application. So we administered these 3 different communities to subjects in a random order, and we also randomized whether shape or color was actually the reliable trait. And which colors and shapes were associated with being good or bad, just to control for any biases. And our results indicated that as richness increased from 2 to 4 to 8, subjects switched from using the reliable trait to using the key trait. So they went from using shape to using color. So this rejects that second hypothesis I was talking about and supports the key trait hypothesis. So the richer the community, the more easily that imperfect mimicry can evolve. But of course, species richness is just one way of describing the diversity of communities. So evenness also refers to the diversity of communities, and it describes the relative abundance of different species in communities. So if you have 999 elephants and 1 giraffe, you have a very uneven community, and effectively you only have 1 species. So because uneven species effectively have fewer species, we might actually predict that as unevenness increases, it could reverse the pattern caused by increasing species richness. So as unevenness increases, predators should shift away from using the key trait and return to using the reliable trait because there's effectively fewer species. So we designed additional prey communities that had perturbed species evenness. So we took the medium richness community and the high richness community, and we just shifted the relative distribution of shapes in them. And we folded those into our experimental design, and we found that in the really rich communities, we did see the predicted reversal. And so subjects shifted from using the key trait to using the reliable trait more. But this didn't happen at these communities of intermediate richness, so subjects' behavior didn't really change very much. So this actually rejects our hypothesis because it should be true across all these different levels of richness. So what's actually going on? I think that what's actually happening is that in a really rich community where everything is really evenly distributed, there aren't very many exemplars of any particular prey species. And so predators have to use a key trait like color or like shape to make discriminations. But by contrast, in those communities of intermediate richness, or in those high diversity or high richness communities that are really uneven, there are some prey that are much more abundant than others. And so predators can just focus on what they want to eat and not really have to learn as much about what to avoid. So basically, what all these abundant prey are serving as is alternative prey. So predators are focusing on these alternative prey and not really learning to discriminate between the members of these mimicry complexes. So to conclude, I'd like to just hope that I've persuaded you that receiver psychology and prey diversity are both important for determining the form of mimetic signals, and also that by integrating community ecology with signal evolution, we can build new and interesting predictions that perhaps marry some of the community ecology literature and the evolution literature. So with that, I would like to thank my co-authors and lab mates who were around while I was developing this, my funding sources, and thank you all for your attention.
We have time for a few questions.
Yona.
Thanks. Thank you very much. Really nice talk. So I have one question. So you showed that imperfect mimicry is more likely to evolve in rich community.
Mm-hmm.
So why then we see all those beautiful examples of perfect mimicry in tropics?
So that's a really good question, and I think that there are a lot of different answers to that. One is that we don't know, like across the whole community, what the distribution of mimetic precision is. And I think someone maybe sitting a few feet away from me may or may not have ideas about that. But also, it could be that looking across the entire community might be misleading because, of course, what really matters for the predator is the community of prey that it's foraging on. And so if you have a really specialist predator, maybe it's just focusing on particular butterflies, and maybe its effective community is actually not that speciose.
Yeah, I agree. And maybe we have just focused on those beautiful species.
Right.
Maybe there is actually more imperfect mimics also available. We just haven't investigated those.
Yeah.
Any other question?
Thanks, this is a great talk. I'm sorry if I missed it, but can you say something about, or say more about how much the people were incentivized to try and collect everything?
Oh, yeah. So about the design. So
them exploring.
yeah. So I'll just go back. I'll flip back to this here. So they were presented with a training board that had these 36 shapes on it, and they were told that they could attack half of them. So they weren't exhaustively exploring the environment in each iteration. And then after we were giving them points and a life bar that went up and down during this game. So their goal, sometimes subjects don't always pay attention to points, but if you give them a life bar, that really, and we also played a really like obnoxious gong noise whenever they got something wrong. That might have been the best incentive actually. But then after they'd trained, then we told them, okay, you have a bonus round, and you're not going to get any feedback during the bonus round until it's all over. And just click on the shapes that you want to attack or that you think are good. So we gave them like kind of like a smorgasbord of different prey that they could attack in this bonus round, and that's what we actually used to make our inferences.
Right, so at that point, actually, they are sort of, they could possibly choose everything.
They could choose everything at that point, right?
Thank you.
Thank you, David.
Oh, no, that's glasses.
Okay, good afternoon, and thanks for coming. Well, introduced predators are a particular case of biological invasions, and they are considered to be one of the major causes for species decline and biodiversity loss. And recent studies have suggested that these predators cause more severe impacts to prey populations than native predators. And this is because alien predators can create new ecological contexts in which the anti-predatory responses of prey may suddenly lose their adaptive value. Uh, because the responses of animals to predation risk have evolved in specific habitats in which prey have coexisted for a long evolutionary time with predators. Prey are innately equipped with morphologies and behaviors to cope with local predators. But, however, in this new ecological context, prey may lack a specific anti-predatory strategies to cope with alien predators, with which they lack a SART.
So our next speaker is Muriel, coming from Michigan State University, and we will talk about anti-predatory behavior in stickleback. Thank you, Muriel.
Thank you. Thanks for having me. Thanks for coming and listening. And I'd like to thank Michigan State University Postdoctoral Association and the ESEB Equal Opportunity Grant to make it possible for me to be here and tell you about Icelandic sticklebacks. So when we talk about climate change, it's usually about its disruptive consequences, and rightfully so. But big climatic events can also create new habitats and new niches, such as new islands or lakes that can be colonized by all sorts of organisms. I don't have the chance to work in such tropical areas, but I will tell you instead about the Icelandic climates. And you might know Iceland for its volcanoes and thermal activity, but also beautiful glaciers and impressive lakes at the bottom of these glaciers. And glacial lakes typically have this beautiful turquoise color that's caused by the ice melting in the water, and all the particles make it a very turbid water with this beautiful color that you can see all the way to space. So from satellite pictures, you see up there the color of this glacial lake. The water is so turbid that if I put my hands in the lake, I won't see my fingers. Here's our amazing field technician last year who had to put on a dry suit and go in very cold water to try and find a trap that we lost in there. He never found it. It's really turbid. When I was preparing this, I realized that sadly, if I increase the map a little bit here to the left, you can see this that you might have seen in the news as you were on the way to this very congress. And on Sunday, the Icelandic government threw a funeral to the first officially dead glacier. Sorry. With a letter to the future to promise to do better, but I'll get there. Glaciers are melting, and not on this picture, but often as they melt and retrieve, they will create new lakes that are then invaded by organisms, one of them being tree spine sticklebacks. Sticklebacks are a very visual species, so they have very well developed eyes, and they use them to find prey items, to find mates, and to avoid predators. Some of the predators they'll find in these Icelandic lakes are trouts and Arctic char. And so one of the things we're wondering about is how do they do this in an environment that is so turbid that they can't possibly see very far? And one hypothesis that we're testing is that if they can't rely on their vision very much, they're going to develop their noses and lateral lines from mechanosensory information. They're going to develop those senses better. So to test this hypothesis, we're sampling all across the island, and we're comparing different type of habitats. I have a color coding for the different type of lakes here that I will keep all throughout this presentation. We're sampling marine populations that are in the ancestral population of stickleback. We're sampling low elevation spring-fed lakes that are sometimes still breached by seawater, so we're expecting stickleback in those lakes to be more closely related to ancestral marine populations. And then we're sampling high elevation lakes that are more isolated from the sea. Some of them are spring-fed, and they're so clear that you can stand on a boat in the middle of the water and see all the way to the bottom. And others are those glacial lakes that are very turbid. And so we've done so for a few years now, and today I'll show you results about 15 of those populations that were, sampled all around the island. We're trying to sample a high variety of watershed and different potential invasion routes from the sea. And you'll see those purple low elevation lakes, marine sites, and here in the middle are the highland sites where we're trying to sample both freshwater clear lakes and turbid lakes nearby. And so we're measuring sensory organs on those fish from all those populations. We're staining neuromass here to quantify how good their lateral lines are working. We're measuring epithelial tissue to have an idea about their nose function. We're sending eyes to Idaho, where they look at both eye morphology and gene expression to get an idea of how good their vision is. And we're sending brains to Texas in the Huffman lab, where we're looking at brain morphology of the different parts to get an idea of how developed the brain parts controlling those different sensory organs are. What I'm going to concentrate on today, though, is behavioral trials, anti-predatory behaviors. So very similar to what we heard before, but we're trying to compare how does fish react to different cues of predation, and depending on where they come from. So we're using two different cues. One is a physical predator, so they can both see it and sense it in the water. You'll see it in a minute. And one is olfactory alarm cues. So these are cues produced by the skin of the fish when they're in danger, pheromone-like cues, so the fish can smell it and know something's wrong, but they don't see or feel anything. And so each fish goes through both cues, both trials in random orders. And for each population, we're testing them in clear water. So that's a full light spectrum trial, and in water that simulates glacial visibility. So we're using UV filter that match actual measurements of light intensity in those glacial lakes. So in those trials, we're expecting the fish to have a much harder time seeing the fake predator. And here's how it looks like. So we have a tank. There we go. I'm not sure if you had time to notice the stickleback here. We're waiting for it to be at a particular position. Push a button. This predator is rushing always the same speed in the tank. We're going to measure the first reaction. This particular fish froze and didn't move. So for the first 2 minutes, we're looking at what they're doing. And for the next 8 minutes, we're measuring activity, where we're going to see how many times it crosses the lines. They have refuges here where we're checking if they hide as well. Yeah. And we've done so for 15 populations so far, nearly 400 fish and 700 trials, and good representations of the different habitat types. So I'm going to start to show you the first reaction to these different types of cues. So here in yellow, you have olfactory cues, so they can only smell a predator, and in red, you have this visual mechanosensory, so the rushing fake predator in the tank. And the first thing you can notice is they react differently to those different types of cues. So they're much more likely to do what we call a fast start, so that it's explosive swimming behavior to escape a rushing fake fish in a tank. And when it's odor, a lot of the time we don't observe reaction, or they will freeze on spot, which kind of makes sense. If you get a signal that there is danger somewhere but don't have the corresponding visual cue, you don't know where the danger is coming from, you just freeze on spot and you try to identify the source of danger. If we break that down, now we're looking only at the reactions to this physical predator in the tank, and they're classified by habitat type. Here you have the 4 different types, and by light treatment here. So we can see again, this light blue is this fast start, so quickly swimming away from the predator. It's more prevalent in marine and lowland spring-fed lakes and habitats. And also something that's really interesting is if you look at the difference from the spring-fed, so full light where they can see everything, to this simulated turbid environment, you have a big increase in the proportion of fish that are freezing, even though there is a rushing predator. So potentially, because they can't see it as well, they'll also start by freezing instead of swimming in some random direction. Now we're looking at reaction distance, and this is the distance, we're measuring the distance from the fake fish and the nose of our subject fish. And this tells us how quickly the fish escape or react. And so a short distance here means that they only react very last minute. And here, from this graph, what I can tell you is fish from glacial environment, this turbid environment, only seem to detect the predator last minute, so they're slower to do so. And this is activity. There's a lot on this slide, both the cues and both treatments. I just wanted to show you the full data set here to tell you that I have a three-way interaction. So there is how much activity the fish have after this predation events depends on the cue we're using, depends on the light treatment, and also depends on where the fish is coming from. So where they come from matters for how they react to those events. And if I break it down, just showing you the mean activity here, they're generally much less active after a rushing predator, which we take as, okay, they've been really scared, they hide somewhere, and they take much longer to recover. But this is much stronger in the spring-fed light treatment. So when they don't see the predators as well, they're quicker to recover. And if we look at it broken down by habitat types and light treatment, what I want you to take from this is it's the same general pattern except here. So those fish from glacial lakes seem to have much increased activity after a rushing predator, but only in this turbid environment that they're supposed to be used to. So to conclude, I'll say that we observe differences in how fish react to simulated predation, and there's differences depending on where those fish are from. And despite some of those lakes being very young and having been colonized quite recently, highland glacial fish seem to stick out a number of times, are slower to react to a predator, less likely to use explosive escape, and more likely to recover after exposure to a predator. And so I don't really have time to tell you much about it, but we're currently exploring what's going on in the head. So we're going to look at both brain morphology and also gene expression of fish that have been through all those trials. And we want to try, and of course with all the sensory morphology, we'll have a better idea of how good their senses actually are, and if this is potentially a change in strategy or actual differences in their morphology and sensory organs. And I'll skip really quickly to thank everybody that's been involved. Maybe? Nope. Thank you.
Thank you, Maria. So now our next speaker is Caleb from University of Guelph, who will talk about cognitive ontogeny in sunfishes. Thank you, Caleb.
Thanks. Thanks everyone for coming. I'm excited to tell you about my work looking at the influence of ontogenic niche shift on brain development, and from that trying to infer sort of cognitive growth trajectories, changes in cognitive trajectories through development in fish. So starting with ontogenic niche shift is a phenomenon that is basically ubiquitous throughout all animals, and it's the phenomenon where ecology changes throughout life because of changes in body size. So it's pretty easy for us to understand this in fish. So we have a little young fish here and a big fish here when it's older. When you're young, maybe you're limited to foraging on small prey like plankton, because that's the only thing you're big enough to eat, whereas once you get older, you're then able to forage on smaller fish, you're able to forage on snails or other kinds of macroinvertebrates, you're still maybe able to forage on plankton, so you have a more varied diet, and you can change the things that you're foraging on. Ecology can also change in terms of predation. When you're a young small fish, you might be extremely prone to predation from size-limited fish predators, like this little shark. Whereas once you get bigger, you then are able to more easily defend yourself from predation, and so you don't have to be as worried about predation. So people have looked at differences in ecology and how that impacts cognitive ability and brain size on a spatial scale quite heavily, and so we've looked at that within species and between species and seen that ecology has a big influence on brain size and on cognitive ability on a spatial scale. But I'm interested in how ontogenic niche shift and these changes in ecology that occur throughout life can influence the growth trajectories of brains, and from that inferring the changes in cognitive ability. So why hasn't this really been answered before? So the reason I would argue is because when organisms grow, their brains, and when fish grow, their brains are changing for non-ecological reasons. A fish that weighs 20 kilograms is going to have a bigger brain than it did when it was a baby, just because you need a bigger brain to control a bigger body. And so these, what I call background changes, developmental changes in brain size, are going to be confounded with any potential ecological influences that you're seeing throughout life. So how do we solve this? And I would argue that the best way to do it, or one way to do it, is by looking at in a single group of fish that have the same background developmental changes, so the same species, but you introduce a split in their ecology into two different ecological groups throughout life at some point in their lives. So the ecology gets different, but the background changes are the same, which allows us to isolate the effect that the ecology is having. This can be done experimentally by introducing different ecological situations at some point in the middle of their lives, but that limits your ability to test sort of ecologically realistic cues. And so the other way to do this is by using a natural population that diverges in its ecology at some point in its life. So from the hypothesis that ontogenic niche shift is partially causally linked to the changes in brain size that you see throughout development, you can then predict that if you have a system where you have ecology consistent in the same group but then diverging, that you would only see divergence in brain morphology or brain size once that ecological divergence has occurred. So the system that I'm going to be testing is with pumpkin seed sunfish. I'm sorry to disappoint the people who thought I was going to be talking about big ocean sunfish. So pumpkin seed sunfish are native to North America. They're a freshwater ray fin fish that typically inhabits these kind of shallow water, what we call littoral habitats, where you're near shore, there's lots of vegetation around you, and they're specialized to feeding on macroinvertebrates like snails and insect larvae. In certain lakes, though, when there is no strong pelagic competitor, they can move out and colonize the pelagic niche, clustering usually around shallow rocky shoals. So if you've ever been boating on a lake and you see a buoy that's marking a rock that you don't want to go over with your outboard motor, that's basically where these fish are, out in the open water. So they're not actually deep, but they are out in the open water. There's much less structure other than the rocks, and they're foraging almost entirely on zooplankton. This system is really good for testing these questions for two key reasons. The first is that though we have these big ecological difference in the adults of the fish, who spend their entire lives out in these two different habitats, the juveniles of the fish, even though they are living in the separate habitats, have very, very similar ecological conditions. So the juveniles are very limited by predation avoidance, so they have to avoid bass, they have to avoid lake trout. And so they stick mainly to the benthos, to the bottom of their habitats, just hiding among the rocks, hiding among the sticks. Both of them are too small to feed on macroinvertebrates in the littoral habitat anyway, so they're all feeding on plankton regardless of where they are. And so we have these very, very similar ecological conditions until the fish reach about 70 millimeters standard length, when the littoral fish are freed from the constraint of foraging on the little fish, and they're freed generally from the constraint of fish predation, and so they can make full use of their littoral habitat. The second reason that this is a useful system is that we found last year that the adult fish in the habitats have very different brain sizes, suggesting that there are different cognitive requirements of the habitats that they're living in. So we found that for the same size fish, fish living in the shallow water have about an 8% larger brain that we linked to the foraging ecology that they have in their systems. So we would expect that even that though they have differences in brain size as adults, because their ecology is very similar as juveniles, that their ecology wouldn't their brain size wouldn't diverge until we see signs of ecological divergence between the groups. Now, we assess ecological divergence using a morphological characteristic of jaw size. So in the littoral habitat where they're feeding on snails, you need a really big, robust jaw in order to bite down and crunch through snail shells. Whereas in the pelagic habitat where you're just eating Daphne, you can just sit there and, oh, there's one, you can just eat it. You're not limited in terms of your jaw. And so we use the ratio of the jaw size to the body size as an indication of when ecology is diverging between the groups. So what we did is we went out, this is Ashby Lake where I did my sampling, collected sunfish of all sizes from various littoral and pelagic sites throughout the lake, aged them by counting annuli on scales, measured their jaw morphology, dissected their brains, and weighed their brains. And okay, what do we find? So this is a figure showing relative jaw size on the y-axis across the ages. And what we found is that jaw size doesn't become significantly larger in the littoral habitat until the fish are 3 years old. And interestingly, 3 years old is also the point when they reach 70 millimeters, which we understood from previous work was what freed them from the constraint of predation. So all being consistent with our expectation that that ecology is similar between the groups as juveniles and then diverges. So from this, we would predict that we won't see any difference in brain size between the fish until they're 3 years old. However, this is showing age 0+ fish. So these fish are 2 months old at very most. And what we see is a difference in what we infer to be brain growth rate. So we see a difference in the interaction of habitat that they were caught in and standard length, influencing brain size. So we have brain size on the y-axis, body size on the x-axis, and you can see the red dots show fish that were captured in the littoral habitat, and the slope of that line is steeper in the littoral habitat, suggesting that even when they're only 2 months old, their brains are already growing faster despite this lack of ecological difference that we found. And this difference in brain size is then continued throughout life. So I'm showing 2-year-old fish and age 4 and above fish here, because we didn't have that many 5 or 6-year-olds. And there's no difference in brain growth rate, but there is a difference in brain size, showing larger brains in the littoral habitat throughout all of the ages. And this occurred for age 1 and age 3 fish as well. So again, we expected that when you see a divergence in ecology, only then would you then see the change in brain size occurring, but this isn't what we found. In fact, we saw a difference in brain growth rate, brain morphology from the very beginning of life. So this leads to some interesting conclusions. So the first one is we don't know exactly what the proximate mechanism generating the difference in brain size is between the groups. It could be genetic or plastic or maternal effects. There's a lot of different possibilities, and we'll be testing that in the future. But what this suggests is that whatever the mechanism is generating this difference, it has to occur very, very early in life, because these fish are on the particular brain growth trajectory that they'll be on for their entire lives from 2 months old. So this could be genetic, it could be some kind of very early life plastic effect, it could be maternal effect, but whatever it is, it's occurring very, very early in life. Second is that it appears that the ecology of adulthood is more important for shaping brain growth trajectories than the ecology of childhood, for lack of a better way of putting it. So it does appear that not all stages of development are equally important for shaping the brain growth trajectories of organisms, and that selection could be acting much more strongly on particular stages of development that are important for shaping success and foraging success and cognitive success in these organisms. So with that, I'd like to thank Rob McLaughlin and various members of the Robinson McLaughlin Labs, as well as the people up at Ashby Lake who housed us and fed us. And then I will take any questions that you have.
So we have time for question. We have one question here.
Thank you for a lovely talk. I just have a quick question about the age effects on. Sorry, I can't sit down. On the brain size, particularly. How can you tell that it's differences in ontogeny versus differences in mortality if you're not following fish longitudinally? I.e., that you have differential mortality for brain size or ecology in the different populations.
Oh, it could be. It could be based on mortality, but whatever that influence is occurring, again, is occurring due to the effects that we see later in life. So we don't see any reason, any ecological reason, to think that there would be differences in mortality based on brain size in the early life. There could be, but they're not evident based on the ecological situations that we're familiar with in this system.
Yep. We have one question here in the front, then second one.
I wonder whether you know that the difference is in volume versus number of neurons.
It's a very good question. So we measured brain size by brain weight in this situation, not specifically volume. But yes, the number of neurons is an important characteristic of the brains, and we haven't measured that yet, though that's something that we would like to do.
We had one more question on the top.
So you compare brain size relatively to body length. So my question is, what if the fish differ in body shape? What if the body length is not the same measure? So have you compared it to body mass or some different measure of size?
Yeah, so we do measure body mass as well in these fish, and body mass is used quite a bit in interspecific studies of brain size to compare, but so the first thing I'll say is that brain size, body length and body mass were really highly correlated, and so our results didn't really change whichever one you used. I think that body length is a bit of a better measure to use here because it's less susceptible to kind of short-term variation in condition than body mass would be.
Thank you, Gareth. So our next speaker is Stephanie from Stockholm University, who will talk about mosaic brain evolution hypothesis. Thank you, Stephanie.
Okay, thank you. So I'll just dive right in. As we all know, there's a huge variation in brain morphology both across and within species. And this is just one of the common figures that illustrates such variation. So one of the reasons that such diversity exists has actually to do with how brains evolve. And so at the center of this debate lies two main brain evolution theories. So at one end, we have the mosaic brain evolution, which basically states that regions are free to evolve independently from one another. While at the other end of the spectrum, we have the concerted brain evolution, which says that regions have to evolve in a coordinated manner given interconnectivity patterns. So studies that have actually looked at how brains evolve have mainly relied on comparisons across species, across populations, etc. So the aim of our study was first to carry out an artificial selection experiment and to use this selection experiment to examine the mosaic brain evolution hypothesis. So in order to do so, we decided to use guppies of Pusilla reticulata. And one of the reasons is that, wild populations of guppies have been extensively studied over the years. So we know that guppies can inhabit both high and low predation sites, and depending upon predation regime, it can actually affect behavior, morphology, brain anatomy, and several other traits. So guppies are also live-bearing fish that actually have a relatively short generation time. So it can take approximately 5 to 6 months for one generation. And they're also relatively easy to keep in the lab. So this is a picture of our lab currently where we house thousands of fish. There have also been previous successes using guppies as a study organism. So in this study by Cotteshaw et al., the authors were able to successfully up-select and down-select relative brain size in guppies. So there was approximately 11 to 14% difference between the large and small brain lines just after 3 rounds of selection. So this shows a dorsal view of guppy brains. So on the left, we have the male. The sign supposed to be here. So this is the male and this is the female. And the region that we selected for is the telencephalon, as highlighted here. So we specifically chose this region because it's known to be a cognitive center, not only in guppies, but in vertebrates in general. So what this means is that this region is actually responsible for a wide range of cognitive abilities. So how do we do this selection experiment? So we have a starting population, F0, whereby we have 3 independent replicates of 75 breeding pairs each. So we put the pairs together, we allow them to produce offspring, and once a certain number of offspring is produced, we then measure the brain morphology of the parents. So using these combined residues, we then rank them accordingly, and we take the top 15 pairs to form the up-selected line here, and the bottom 15 to form the down-selected line. In this particular selection experiment, we also included a control line. So basically, we just took 15 random pairs from the starting population, and we used it to propagate the next generation. So okay, in generation F1, we have 3 replicates, and we have 30 breeding pairs each in the up, control, and down selected lines. So similarly, we allow a process whereby we just allow them to produce offspring, and then once a certain number of offspring is produced, we then measure the brain morphology of the parents again. So this is a similar process as the previous generation. So this goes on. But today I'll be talking about generation F2, so what happens after 2 rounds of selection for telencephalon size. So as I mentioned, we have the breeding pairs, and we euthanize them in an overdose of benzocaine, and we fix the entire fish in 4% formalin for a week, after which we use a dissecting microscope in order to dissect out the entire brain. And from each individual brain, we then take images for 4 separate views. So basically the dorsal, the ventral, and the 2 lateral views. So based on these 4 separate images for each individual brain, we then use image J in order to quantify brain morphology using the ellipse submodel as shown by the formula here. So in addition to telencephalon, we also measured several major brain regions such as the optic tectum, the cerebellum, the dorsal medulla, the hypothalamus, and the olfactory bulb. So today, the results that I'll be presenting has been obtained after dissecting approximately 1,500 brains. So what have we found so far after 2 rounds of selection? So first I want to mention that I'll be showing proportions just for visualization purposes, but if I were to use absolute volumes, the results show a similar trend. So here on the x-axis, we have the selection lines, and the y-axis, we have the telencephalon. So for females, we see the up-selected lines in blue here. It has significantly larger telencephalon in relation to both down and control lines. So for males, it shows a similar result whereby the up-selected line has a larger telencephalon in relation to the other 2 lines. So from this result alone, it seems that it may be easier to up-select rather than down-select for telencephalon. Now, what are the correlated changes that occur in response to directed selection for telencephalon? So here on the x-axis, we have standard length. On the y-axis, we have the overall brain volume. So we see significant overlap across the selection lines for both females and males. So what this indicates is that there is no significant difference in terms of relative brain size across selection lines. Interestingly, however, we find a change in another brain region, so specifically the optic tectum. So this is the region highlighted here. Again, on the x-axis, we have the different selection lines. On the y-axis, we have the optic tectum. So looking at females first, we see the up-selected lines has significantly smaller optic tectum in relation to both down and control lines. As for males, however, this effect is not as pronounced, but we see it going in a similar direction. So what this seems to suggest is that by up-selecting for telencephalon could actually lead to a correlated decrease in the size of the optic tectum. So we did not note any other significant changes in any of the brain measures that we took. So we see this difference in brain morphology in adults. Are these differences already present at a young developmental stage? So to do this, we took pictures of juveniles from the top view. So you can see the head is semi-transparent, so we're able to obtain or measure the width of the telencephalon and the optic tectum here. So we use this as a proxy. So the ratio of the telencephalon in relation to the optic tectum, as shown by the axis. So we see no significant difference across selection lines. So we wanted to see how is this difference present in adults? So how does this relate to our adults? So we took similar measures, and here again the similar axis, and we see that the up-selected lines, as highlighted in blue for both females and males, has a significantly larger telencephalon to optic tectum ratio. So this seems to indicate that such differences in brain morphology actually arises later on in development. And as we all know, neural tissue is highly costly to maintain. So we wanted to see whether there are certain trade-offs of cost involved in such selection. So to do this, we quantified reproductive output across our selection lines. And we see here, so x-axis again selection lines and y-axis the total number of juveniles. We find no difference in terms of total number of juveniles produced across the selection lines. So how does our selection lines actually relate to what is seen under wild conditions? So for this, we compared with wild guppy brains. So we had wild guppy images of wild guppy brains as well. So I measured them, and here we see that wild guppies actually have significantly larger telencephalon in relation to both down and control lines. So this could be due to a domestication effect whereby our lab-kept guppies that have been kept in the lab for several generations have an overall reduction in brain morphology. So it seems that our up-selected line actually closely resemble what is seen under wild conditions. So after 2 rounds of selection, we actually find support for the mosaic brain evolution, whereby it appears easier to actually up-select rather than down-select for telencephalon. So as I mentioned in the previous slide, it could be that we are actually selecting for our telencephalon to return to how it is under natural conditions. On the other hand, it could be there is a minimum requirement for this particular brain region in order for guppies to actually sustain their normal functions. So there could be a certain floor effect on how small this region actually can be, which makes it more difficult to down-select rather than up-select. So we also find a correlated response in the optic tectum. So specifically, by up-selecting for telencephalon, we find a correlated decrease in the optic tectum. And one of the hypotheses could be due to a shared progenitor pool of cells, whereby these cells could actually differentiate into a telencephalon or the optic tectum. So there could be this certain trade-off between these 2 brain regions. But this hypothesis requires further testing on a molecular level in order to understand what actually is going on a final scale. So these are the assays that we're planning to do in this selection lines, and we'll be starting with reversal learning, visual acuity, but we also hope to do several genetic analysis in order to see whether gene expression changes in the selection lines and how heritable are these traits. So this is a growing list. And at the moment, I am currently in generation 3 of the selection line. So we hope to see how this selection actually progresses. And with that, I'd like to thank everyone involved in the project, the funding, and thank you for listening.
We have time for question. Yeah, Ruben?
I wonder what you know about number of neurons in your different lines.
Yeah, I mean, There has been a paper recently published about the neuron number in the large and small brains. So it appears that larger brains actually have more number of neurons compared to the small brains. But we don't know yet because these lines have been established very recently. So we haven't done much tests. So now is where all the testing begins.
Other question? Yeah.
When you were selecting for the high and low telencephalon lines, were you doing it by raw telencephalon size, or was it relative telencephalon size that you were selecting for?
Relative. Yeah. So it's the telencephalon in relation to the rest of the brain.
There is one question here in the center.
Thank you for your talk. I find very interesting that you find that the one you selected for the increase were the most similar to the wild ones. Do you plan to do also the battery of tests on the wild ones to see if they match your selection one?
Not at the moment. I mean, we don't have those wild fish. I mean, we euthanize them to quantify their brain morphology. But I mean, that is something we can do in the future. But I mean, currently we just hope to do with these lines to see whether there's a difference in performance across the lines first, and then we can compare them to the wild.
I think that would be really interesting because all the cognitive tests are mainly done in captivity, and then if you can show that the difference between the wild and the one you manipulate are what comes there would be really, I think, very relevant for many fields.
Yeah, definitely.
Other questions? Yeah, second one here for Caleb.
So I'm wondering how your interpretation of the sort of correlated change in the optic tectum along with the increase in the cerebellum and the telencephalon, and the decrease in the optic tectum, do you interpret that as further support for the mosaic hypothesis, or does that correlated response actually indicate some sort of concerted change, even if it's in the opposite direction that we might have thought?
I mean, it's not really in the opposite direction, but I mean, we show that it's able to actually select for a specific brain region, but we also expect like changes in regions that are interconnected. But it shows that the relative brain size doesn't change even though we select for telencephalon. So that this is also support like there is mosaic brain evolution going on, even though there's changes in optic tectum as well. So it could be this is also a mosaic way of changing, but there is still, it's obviously not only mosaic going on. There's always going to be a balance between the two hypotheses.
We have time for a last one question, I guess.
Can you refine your analysis to see what happens within the telencephalon? Is there a specific part that increases?
What?
The telencephalon is relatively large. Can you refine your analysis to look within the telencephalon to see what changes have occurred?
I mean, yeah, not at this point. I mean, we're just using this method to see like based on how we have selected previously. But yeah, in the future we hope to do scanning to see how neurons or interconnectivity patterns changes within the structure itself.
Thank you, Stephanie. So our last speaker for this symposium is Marcus from Turku, and he will speak about annual predation risk and brain size. Thank you.
Thank
Marcus.
you. Well, let's go out in the wild this time. And I want to start by acknowledging my partner in crime, Kimi Aatinen here, trying to approximate his brain size here. All right, so the evolution of brain size has been viewed as a trade-off between energetic constraints and natural selection for enhanced cognitive abilities. And so under this scenario, the threat of predation should select for greater investment in neural tissues that help animals evade predation. So that's why we should expect a positive relationship between predation and brain size. So case is now closed and I can step down from the podium, I guess. Well, hold your horses, not quite yet. Yeah, there's more to it than meets the eye. And in fact, when we look at recent literature, it has challenged the view that predation should select for larger brains, often finding even the opposite pattern. So in fact, evidence for positive effects of predation on brain size is quite rare in nature. So what's going on here? Is it just that the people are studying fish are very confused and fishy, or what is it all about? Well, I think that the kind of part of the solution might be that the landscape in nature is more complicated than we currently think. For example, there's fluctuating selection going on, and that might kind of slow down the rate of evolution. And second, we often tend to forget that there are other costs than physiological ones in evolving a larger brain, and these can relate to the decision-making process itself. So let's start with a really simple question: are bigger brains faster than small ones? And well, you think that even a small child can answer this: yes, they have higher computation capacity. But when you look at the empirical evidence, it's often so that big brains are coupled with slow decision making. So what's going on here? Well, I think that the speed accuracy trade-off might be one part of this riddle. So we all encounter speed accuracy trade-offs in our everyday life. So for example, when you try to enter, can open your home door, if you are too fast, you won't succeed. So there's lots of speed accuracy trade-offs going on, and usually if you take more time, this error rate goes down. But if you have a complex task, that will take longer to solve. But if the cost of making wrong decisions is not too high, you might just make a quick guess. So avoiding these mistakes during a task requires time, but also cognitive capacity affects its likelihood of making correct decisions. And then if you for example, in predator situations, it's often a question of life and death, and here errors are severely punished. And what I argue here is that individuals with different cognitive capacities, they might focus on either long-term or short-term benefits. So the big brainers here, eventually they might be able to reduce the error rate of their decisions, so it may pay for them to take a bit longer with the decision and surely get it right. But if you're dumb enough that it doesn't help you much to hesitate, just make a quick guess and save lots of time. So the logic here goes like this. So if the error penalty of errors increases, for example, when there's an increase in predation risk, then you would expect accuracy to be favored over speed. So this should be the advantage of the big brainers. However, the situation changes under benign conditions when it's often better to make a quick guess and save lots of time, and time is an important fitness consideration in many animals. For example, in breeding sea ducks, breeding success goes down dramatically with the season. So is this just pure science fiction? Well, perhaps not entirely. So when we look at our own data with eider ducks, these big brain females with relatively big brains, they breed later. And also, if you have a kind of high predation year, as in this graph over here, we can see this vertical blue line. That's the kind of head size of those females losing their nest to predation in this really benign year. And we can see that it's significantly larger than in the population at large, based on simulation from the population. So what we did here is to hypothesize then that large-brained females might then have higher survival during these dangerous years, whereas the opposite might be true in safe conditions. And we also came up with a clever mechanism, because it's been shown recently in waterfowl that if you have kind of severe predation episodes, then predation occurs quite rapidly during the season. Whereas if you have more kind of lower predation risk, there's a peak in predation just towards the end of the season that might then disfavor these late breeding big brain females. So we tested that mechanism, and we also validate this head size, brain size relationship by looking at a nice little sample of fresh Danish skulls of female eiders. And here we have this study area. It looks very idyllic, very nice. However, I can smell a rat here. So if you're an eider female that wants to select a place where you are surely to get killed by a predator, then you should select our study area. It's kind of the survival of female eiders here is the lowest ever recorded. And our data here kind of consisted of data on females where we had complete breeding data and head volume measurements, and we then found these guys killed at their nest. And we measured predation risk just by dividing the total number of killed females at their nest by nesting attempts in the area. And these recovery dates of the female carcasses, that was used to kind of construct an annual cumulative frequency distribution of the seasonal timing of predation. And we made two analyses here. The first looked at survival in relation to head size and candidate predictors, and we always forced structural size into these final models, because we were kind of interested in the effects of relative brain size on survival. Then we also had a look at the seasonal timing of predation, so how this cumulative proportion of females of all kills for that year, how it changed according to annual predation risk and the timing within the season and their interaction. Finally, some results then. So here we can see kind of a safe year and a dangerous year kind of. So we find an interaction really between annual predation risk and the head size. So if we have a kind of safe year, not so much predation, we can see really that the small-headed females had higher survival than the big brainers, whereas the opposite was true in this dangerous year. They're kind of the large brain females out-survived these small brainers. So that's according to our predictions then. And what about this mechanism then? Well, from this graph, we can see that in kind of this cumulative proportion of killed females, of all females, that increases more steeply during the early season in these dangerous years, whereas there's more of a peak in predation towards the end of the season during these low predation years. So that was also according to what we predicted. So the implication of all this is that if you have a dangerous year, it will be dangerous from the beginning. Whereas if you have a safe year, then kind of this predation will more happen towards the end of the season. So females breeding late in the season are kind of more severely affected. So to conclude, I hope that at least our data suggests that there's kind of brain size specific breeding phenologies, and these may, under variable predation risk, give rise to fluctuating selection on brain size. And we think that this is a bit unexpected result is due to this big brainers being disproportionately hit by breeding later in the safe years. So neither this fast or slow strategy is consistently superior over the other. It's context dependent. And the good thing here is that this explanation is quite simple, so it doesn't require that these big brainers would be kind of better at as escaping predation. But what we don't know at the moment is how common these brain size specific breeding phenologies are in other study systems. So that's a question that we really would like to look at in the future. And with that, I thank you.
Any questions? Ruben?
You chose to focus on brain size rather than relative brain size. I wonder why.
Yeah, we looked at the relative brain size. Yeah, because we think that kind of if you have an animal that is bigger, there's a very close correlation with the kind of brain size, but it doesn't mean that this female would be kind of more clever. It's just that you need a bigger brain to kind of operate a bigger body, I would say. We had absolute brain size and kind of this relative size in the same equation, so in that way it became kind of relative brain size. Yeah.
Yeah, one question here.
Yeah, we have also measured kind of the same things with the ducklings as well, and it seems that of course we don't know the pedigree how much it's relatedness, but still, if you have a kind of big brain mother, the ducklings will also be on average kind of big brain there. So there's correlation there. Yes.
Another question?
No questions about neuron numbers or nothing. No.
I just have a quick question. Yeah.
Yeah.
Why would a big brain size female would be more exposed at the end of the season, even if you are breeding late in the season?
Yeah.
Yeah, but why are they more exposed than the smaller female?
Because they are kind of more sensitive in their strategy, so they are kind of more choosy and picky before they choose their nest site. And when you have this kind of quite safe conditions, it's much better to start breeding immediately. Yeah.
Yeah.
Regarding choosing the nest sites, what time span are you looking at between choosy, picky females and quick decisions?
Yeah, it's on average about 8 days, so that means 1/3 of the complete incubation period of these guys. That is about 26 days.
Okay, thanks.
Thank you, Marcus.
Yeah, thanks.
Welcome back, everyone, to this last session of the symposium on the evolutionary ecology of aging. And our first speaker is Martin Tyszewski, who's going to talk to us about aging in free-living gratids.
Good afternoon, ladies and gentlemen. I'm a PhD student from Charles University in Prague, and in my PhD I focus on aging in free-living population of gratid. If we should define aging, we may say that's a process where the physiological function and processes typically decreases with the age. But in the same time, the risk of mortality is increasing, however, fertility or fecundity are typically decreasing. Aging used to be quite overlooked process in studying free-living organisms, especially in small passerine, because they have very high extrinsic mortality caused by predation. So we just simply didn't believe that such a small organism may have a time to age. However, this is not true anymore, and now we know that they could serve as a convenient model for biomedical studying of aging. In my PhD, I focus on aging from many different perspectives, but today I will be mostly talking about physiological stress, assessed by immunity, and maintaining of oxidoredox balance in organism. We were just interested whether the level of antioxidants and markers of oxidative stress are changing with the age. And in the end of my presentation, I would like to put all these findings together and test it whether there is any effect of reproduction on increased oxidative either physiological stress. As a model organism, we have been studying free-living population of gratid for almost 9 years. And since that time, we captured more than 600 individuals, and some of the individuals luckily several times. And this dataset of repeatedly captured individuals we use for description of aging on within individual level. In this point, I should mention that that's not always easy to identify accurate age of the birds. So the age included in our analysis was based on the bird ranking as a minimal estimated age. To be an organism in health state, the redox balance in organism has to be maintained. On the one hand, there is an overproduction of free radicals during the cell metabolism in mitochondria, but free radicals are not produced only there, but they are also produced during the oxidative burst. That is a reaction of immune cells against pathogen. And if the level of this markers, if the level of these free radicals are not controlled under strict control, they can really impair the cells and tissue. And since the aging is very often related with the shift in redox balance in organism, and aging is related with increased oxidative stress, we decided to measure the concentration of several antioxidants, and those involves free thiols that possess various group of molecules having SH bound that can be oxidized in oxidative environment, and it means that in this environment they neutralize free radicals. And the second and third antioxidant were the antioxidant enzymes working synergically. First, superoxide dismutase is an enzyme that decomposes very reactive superoxide radical into less reactive peroxide dioxide, and this peroxide dioxide is subsequently breaks down by glutathione peroxidase activity into the water. So first, we're interested whether the level of this antioxidant is increased with the age. However, for total thiols and superoxide dismutase, we didn't observe any effect of age on the concentration. However, activity of glutathione peroxidase was decreased with the age, which indicate that older individuals decomposed peroxide dioxide less effectively than younger individuals. And to quantify how much older individuals suffer from oxidative stress, we measured the concentration of several markers of oxidative stress. And this is quite a new approach in field research, I should say, because from the clinical study, we identified set of markers whose concentrations are typically increased during the autoimmune or inflammatory diseases. And this dataset includes molecules involved in DNA or RNA damage, protein damage, lipid damage, as well as in inflammation. And the concentration of all these markers was assessed by mass spectroscopy. So first, we check whether the concentration of these markers are intercorrelated. And this is what can you basically see in this Pearson correlation matrix, where the blue is positive correlation, red is negative correlation, and the shape of the cloud represents the goodness of the correlation. So what is quite obvious is that a concentration of all these markers of oxidative stress are highly correlated. Then we interested whether they increase in their concentration with the age. And this is what we basically observed for all markers. So independent of sexes, concentration of all these markers was linearly increased with the age, which indicate that older individuals suffer more from DNA damage, lipid damage, protein damage, as well as inflammation, which is quite consistent with hypothesis of inflammatory aging. And surprisingly, age was only one significant factor that explained the concentration of all these markers in our analysis. And okay, so we observed that markers of oxidative stress are increasing with the age. However, glutathione peroxidase activity was decreased. So we asked, is there any causal link between that? And my answer is yes. We found that there is a negative relationship between glutathione peroxidase activity, for example, with leukotriene beta 4, which is marker for inflammation, or with the 8-isoprostane, which is the marker of hydroxylation of lipids. So the individuals that have less active glutathione peroxidase activity suffer more from oxidative stress. And now let's a little bit move up from molecular or mechanistic level to more organismal level. And to assess the physiological stress, we use quite common indicator of physiological stress that is called HL ratio. And this is the ratio between number of lymphocyte and number of heterophile in organism. And the higher HL ratio, the higher physiological stress in organism. And when you plotted residuals for HL ratio from our model against age, we observed a linear increase of HL ratio with the age, which indicate that older individuals suffer more from physiological stress. And then we ask, is there any link between oxidative stress and physiological stress? Because those indicators are quite different indicator of total stress in organism. However, we didn't find any cross-link. The reason could be probably that physiological stress more reflect chronic and acute state of the organism as well as the state of immune system. And it brings me to the last point of my presentation, and this is testing of reproduction on physiological or oxidative stress. We just simply ask whether individuals investing more into reproduction suffer from increased oxidative or physiological stress. And for oxidative stress, we didn't find any link with the reproduction. However, that was not true for physiological stress, where we observed that females having bigger clutch size had also increased HL ratio, which indicated they suffer more from increased physiological stress. And those results are quite well consistent with some previous study done even in gradients, and it also well fits to the hypothesis of disposable soma theory, which predict that there is a trade-off in investment energy or resources either to reproduction or self-maintenance, which is in our case represented by immunity. To put it all into a nutshell, in our study we clearly demonstrated that free living population of gradients aged, as was indicated by increased oxidative stress or physiological stress. Oxidative damage was inversely related with antioxidant activity of of glutathione peroxidase. Investment into reproduction increased physiological stress, but not the oxidative stress. And within this study, we identified a set of markers that could be quite widely used in field research. That's all. I would like to thank all the people who are enrolled in this project, especially to my supervisor, Michal Winkler, who is here, my co-supervisor, Tomáš Albrecht, who came with the idea to study aging in free-living birds, and you for your attention.
Thank you. So, any questions?
Hi, so Sandra, the first panel speaker this morning, mentioned the difference between longitudinal and cross-sectional analysis when you're regarding aging. So is in your data sets, so mostly cross-sectional, longitudinal, or combination of both? And were you able to test for selective disappearance regarding oxidative stress markers?
In the dataset that I presented is based only on repeatedly captured individuals, but we have also dataset of cross-sectional, and we would like to test the effect of different survival. However, this is not yet done within this dataset, but based on the preliminary result, for example, for HL ratio, it appears to be some effect of selective disappearance for some immunological trait.
Any other questions? I actually have one. I was wondering whether, so I guess the individuals you measure from this study are all adults?
Yeah, those are all adults captured in second and older years.
And do you also have data on chicks? Since we saw that early life, Pat's mentioned yesterday, early life environment is very important. So I was just wondering whether maybe you also had data on chicks.
Yeah, I'm afraid that we don't have a complete data to such a hypothesis, because primarily the project was focused on variation on immune genes, and this is side effect of the study where we were maintaining the breeding population of catids.
Okay, thank you.
Hi. Sorry, I may have missed it, but when did you measure the individuals, at which point in reproduction? When did you get the sample for oxidative stress measures?
All samples that we gathered are from during the feeding period. So we capture all the birds when the chicks were between 7 and 15 days old.
Maybe one last question if we have some time. No? Okay, well, thank you again for this presentation. to us about early life effects and thyroid hormones.
Okay, thank you very much for the kind invitation to give a talk. And first I want to acknowledge my collaborators, and especially Binyan, Nina and Antoine for their work, which basically they have done most of the work for this talk. So probably most of you have been to the sessions before. You're familiar with the concept of telomeres, which are key hallmarks of aging. So they are the non-coding sequences at the end of the chromosomes. They protect the DNA coding parts, and they shorten every cell division, and also respond to reactive oxygen species, ROS, which Martin kindly described in the previous lecture. And it has been shown quite convincingly that telomere length is related to lifespan, for example, in zebra finches, and there was also a recent review on this. Also, quite well-known phenomena is that early life environments is a key predictor or influencer on telomere length and aging, as we heard from Pat's talk yesterday. Some critical factors that have been shown to influence telomere shortening and telomere length are early life psychological stress or adversity, poor nutrition, and also prenatal stress hormone exposure. And today I'm going to talk about another type of hormone, which we think it's a key hormone, and it's probably linked to aging, but has been studied much less in wild systems. And this is thyroid hormones, which are triiodothyronide, which is T3, the active hormone, and T4H, the thyroxine, the precursor hormone. They are, as all hormones, they are pleiotropic. They play functions in many different organismal physiological processes, such as development, also timing of migration, molt, reproduction. But today, the most clear link that I want to make is to metabolism. And it has been found in rodent models and humans that actually circulating thyroid hormones levels are associated with lifespan and mortality. So our question is that, how about prenatal exposure to these thyroid hormones? How does it link to aging or aging markers? To study this question, well, first of all, some predictions based on our previous studies. So we have shown previously that exposure to elevated prenatal thyroid hormone levels is linked to increased metabolic rates and also growth, although the patterns have been quite variable. But both these processes could lead to increased oxidative stress, which is the imbalance between reactive species and antioxidants. And when there's more of the oxygen-reacting species or other radicals, then you get damage to your cells. And this can lead to shortened telomeres. Or you can have a direct link from increased growth to shortened telomeres. And then this is linked to lifespan. This is the prediction. And to study this, we conducted a couple of experiments in small passerines. The first species is called flycatcher, with a quite classical, almost like a lab rat of ecological studies. This was conducted in the island of Scotland in Sweden in a nest box population. And the experimental design is very simple. So we have clutches of eggs, which were injected either with elevated thyroid hormones here or control. It was done before the embryonic development starts. And then we followed hatchings from these eggs, collected blood samples just after hatching. And this day 12 responds to just before they leave the nest. So this period here is the main growth period. And we measured telomere from these samples using qPCR, so relative telomere length, and also TRF, so the absolute telomere length. So what did we find? So here we have first the relative telomere length, the qPCR results on day 2 and day 12. And what we find here is that the red bars, the thyroid hormone, chicks from thyroid hormone elevated eggs, actually show longer telomeres than the controls. And the same is found here when they are older, and the rate of telomere attrition is the same in both groups. so slightly against our predictions, but we'll get there. Interestingly, or kind of, we found the same results also with the absolute telomere length. So this is the difference within the two groups on day 12, about 2.1 kilobases. And to give you an kind of idea how much this is, that is actually a significant difference, is that when we look at adult colored flycatchers and their telomere length across years, you can see that this is the shortening rate. So this is about 3 years of shortening, this difference compared to what the adults do. Okay, so some difference here, but in favor of the thyroid hormone group. How about the other species? So here is the graded as the same species that Martin has in his data. So we have an ESEB population. This is from Turku, Finland, and similar kind of experiment. So simply injecting hormone into eggs to elevate the concentrations within the physiological range. That's key for the both studies. And following up the chicks, sampling day 7, day 14. This is the time when they leave the nest. So this is again a critical growth period. And then we also sampled these birds during winter when they were adults to look at the long-term effects of this early life. And here we measured this telomeres using qPCR. And these results, again, here we have relative telomere length, day 7, day 14, and adults. And opposite to flycatchers, there's no difference in their early life or early telomere length, but then they start to accumulate some differences. So here, thyroid hormone elevated group has shorter telomeres than the control, and this actually, the difference even larger here in the adults. So there's actually indeed attrition in the TH group. So what do we find? We have two experiments, two quite similar personalized species, although one is migratory, one is resident. That's something to consider, and they show different effects on telomere dynamics. And how can we explain this? Well, one of the predictions was that it might be related to growth patterns. So just take a quick look at the growth patterns. So here first is the body mass growth of the gratids. And here we can see that early life, there's no difference. Maybe these are slightly smaller, this thyroid hormone elevating group. No difference here. But still, they grow slightly faster here. So they start slightly smaller and gain the same body mass here. So, and if we compare that to our results here, so we had slightly faster growth and shortened telomere here. So actually in this species, the growth pattern fits the pattern of the telomere shortening. How about the other one? Here is the body mass of the colored flycatchers, day 2. So actually our thyroid hormone group birds are larger here at hatch, but they have longer telomeres. And here they catch up with the control group, so they basically grow slightly faster, but here there's no difference in telomere attrition. So certainly these two species show different patterns, and it would be nice to have some explanation. Maybe you can help us with this. But we have one potential hypothesis for the colored flycatchers for this early, really like embryonic stage telomere differences, and that's telomerase, which is the enzyme that can lengthen telomeres. And what we found out is that actually in this telomerase enzyme, in this catalytic subunit, there is a thyroid hormone receptor binding site. So this is human data. We have no idea of birds, but maybe potentially thyroid hormone, prenatal thyroid hormone is actually activating telomerase here, and this is leading to the increased, longer telomeres. So far, I would say that my take-home message from this is that yes, early life thyroid hormone exposure, so variation in thyroid hormone levels, can influence telomere length, also early life, but also later in life, and maybe it has a potential effects on aging. So again, thanks to all my group members and collaborators and all the students that ran in the field and collected the samples. Thank you very much.
Okay, thank you, Suvi. Any questions? Pat?
Thank you. That was very interesting. I wondered if you knew anything about the effect of increasing egg thyroid hormones on later life thyroid levels or metabolic rate, or it must potentially have widespread effects.
Yes. So far, the results I presented that we know from metabolic rates is only from chicks. So no, we don't have, from these birds, we don't have data on like long-term on, and also we don't yet have long-term data on the plasma levels. We suspect that there can be programming of the thyroid axis, but whether it's, we don't yet know whether it's at the receptor level or binding protein level, at what level it is. But this is certainly something we want to look at. Yeah.
Any other questions? I actually have a question for you. I was wondering, so maybe I missed it, but the flycatcher population and the tit population, like one is in Gotland and one is in Bristol.
Mm-hmm.
So could the difference you see at the initial point, could it just be a difference in environments from the two study sites? Maybe.
Yes, they certainly differ in that, like the flycatcher population is, yeah, it's a more deciduous forest type. But there's also this difference in that one is a migratory and one is a resident, so maybe it's the life history differences. Maybe they have different sensitivities for the hormone, for example. And one thing I didn't mention is that we have measured hormone levels in the eggs of these and many other species, and it seems that the levels in great tits are super low, and in this flycatchers they are like 5 times higher. So even the initial differences might tell something about the sensitivity of the species. Yeah.
Anyone else? No? Okay. Well, thank you again for your presentation.
Okay, our next speaker is Sil van Lieshout, and he's gonna Sorry.
That's fine.
Trying. And he's going to talk to us about early life effects on telomere length and lifespan.
Thank you very much. Hi, everyone. Let's start with a bit of background into senescence. So senescence is the decline in performance in older age. And we know there's a tremendous amount of variation between individuals in the way that they senesce. Now, one of the factors that could contribute to this variation, as Pat already mentioned yesterday, is the early life environment. And one of the important reasons for this is that the forces of natural selection are strongest in early life. If we look at the graph, we see survival probability, or you could even see this as the proportion of the cohort that is still alive, declines with age, and then it comes under a so-called selection shadow, where deleterious mutations can accumulate, and that leads to the senescing phenotype. So that means that adverse early life environments can lead to faster rates of senescence. And this has been shown previously in the alpine marmots in terms of social conditions, but also in terms of resource availability and resource competition in the Seychelles warbler and the red deer. So there seems to be a role for early life environmental quality in senescence. However, we should also consider an individual's somatic state and the ability of the individual to anticipate its own future somatic state based on the current environment. And therefore, it can adjust their life history accordingly. And that is described in the internal predictive adaptive response, or internal PAR hypothesis. Now, besides early life environmental quality, we should also consider variability and think of seasonal or even daily variation. It has been recently hypothesized that in these temporally stochastic environments, individuals should adopt a bad hedging strategy, so invest in the soma at the expense of reproduction, and that could lead to slower senescence in variable environments. However, a recent modeling approach showed that that included the declining forces of natural selection with age, leading to slower adaptation of traits that are expressed in later life, indicated that there's actually faster rates of senescence in variable environments. So what we want to test here is, first of all, whether senescence is negatively linked to this early life environmental quality and whether that depends on an individual's somatic state, and whether senescence is positively associated with early life environmental variability. To quantify senescence, we use telomeres, so telomeres are these hexameric repeats, as mentioned before, at the end of eukaryotic chromosomes, and they maintain genomic integrity. And an important trait from telomeres is that they shorten with age due to the end replication problem, and that process can actually be accelerated by environmental stressors. So in that way, telomere length can actually function as a biomarker of senescence and also reflect an individual's somatic state because it reflects the physiological consequences of these individual experiences. So we use these telomeres in a wild population of European badgers in white moods in Oxford in the UK. And it is a population that has been intensively studied since 1987 with a decent trapping efficiency. About 95% of the badgers we recapture within 2 years. So that provides us with full life history data set on all of these individuals. Now, an important trait from badgers is that they mainly eat earthworms, which are only present in moist and mild conditions. So when we actually want to look at environmental variability, we can actually use weather variability as a proxy for the variability in food availability, and use that as a measure of environmental variability. So when we look at our models, weather variability will actually be in there. So the next thing we did is we wanted to get these telomere length estimates, so we caught the badgers, extracted the blood, did some DNA extractions, optimizations, and monochrome multiplex qPCR. So we did the control genes and telomere reactions in the same well. And for this particular study, we use a subset of the entire data set, which is 435 samples from the same number of individuals, for which we have early life telomere length estimates, so within the first year of life. My screen is disappearing. So the first thing then we checked is whether early life telomere length links to life history. And early life telomere length does predict lifespan. However, we saw that this association was mainly driven by conditions in early life and by the relationship between early life telomere length and survival to adulthood, but it does link to life history. However, in this case, we want to look at early life environmental quality. Yeah, it's fine again. A good way to measure the quality of the environment is by looking how many cups survive to the next year. And so we calculate this metric of cup cohort mortality that we adjusted for the trapping efficiency. And to give you an idea of what that looks like, we have cohort on the x-axis and a centered value for that cup cohort mortality to give you the quality of each year, but also the annual variation in quality among years. So then we build a model, and we have cup cohort mortality, so that measure of environmental quality on the x-axis, interacting with early life telomere length as a measure of somatic state. In the model, this is a continuous variable, but here it's shown as a factor, and looked at the effects on lifespan. And what we see is that badgers born in favorable conditions have longer lifespans, independent of their early life telomere length. However, when we look in early life adversity, so on the right bit of the graph, we see that individuals with longer early life telomere length have longer lifespans compared to individuals with shorter early life telomere length. So there is this interaction. Okay, that might be a bit complicated, so let's simplify that a bit. If we compare favorable conditions to adverse conditions, we see that cubs born in favorable conditions have longer lifespans, whereas cubs born in adverse conditions have shorter lifespans. However, we see that in favorable conditions, this is independent of an individual's somatic state, in our case, early life telomere length. But in adverse conditions, when you have a poor somatic state, you have shorter lifespan to individuals with a better somatic state. Now, when this lifespan would be traded off with reproduction, as described under some theories of senescence, this could actually form an adaptive life history strategy that depends on early life environmental quality and an individual's somatic state, as described in the internal PAR. Now, we also wanted to look at environmental variability, and as I said, we used weather conditions and weather variability in the model. So we first will look at rainfall, and we have on the x-axis the mean daily rainfall that interacts with rainfall variability, again, continuous in the model, but a factor for now. And we look at the effects on cup telomere length, so within that first year. And what we see is this negative interaction. So we see that individuals experiencing high levels of mean daily rainfall and experiencing low levels of rainfall variability have the longest telomere length. And the opposite is true for low levels of mean daily rainfall. We also looked at temperature and same kind of interaction, same kind of model. We see that individuals, or cubs I should say, born in colder conditions have shorter early life telomere length. However, these effects are completely mitigated when temperature variability is low. There is, of course, also a correlation between temperature and rainfall. On sunny days, it rains less, as we experience now here. So that might explain why, with high temperatures and high temperature variability, individuals actually have longer early life telomere length, because that high temperature variability also leads to more rainfall. So again, I'm going to simplify this a bit. Same kind of cartoon. Cubs born in conditions with higher rainfall and higher temperatures, which coincide with high levels of food availability, have longer early life telomere length. However, when we add a measure of variability to this, so any deviation from these optimal conditions, we see that they have shorter early life telomere length. And this corroborates a previous study in our study system that showed lower recruitment and lower survival probability when variability was high. And on a longer temporal scale, so in terms of senescence, for example, you should also look at maladaptation. However, in adverse conditions, we see that individuals tend to have shorter early life telomere length, but when we add a measure of variability here, this is actually a temporal increase in the quality of the environment. And this temporal improvement leads to longer early life telomere length. And this temporal improvement may allow individuals to modulate their energy allocation trade-offs, for example, between reproduction and somatic maintenance. So when we look at the effects of early life conditions, we should not only consider the quality of it, but also the variability therein. And in our case, we see this negative interaction between quality and environment of early life telomere length. And I'll end with thanking a lot of people who are involved in this project. I mainly like to thank Amanda and Hannah and Elisa, who are both here at this conference. Thank you very much.
Any questions?
Thank you very much. Very nice talk. So I was wondering, like, so you're presenting only telomeres here, but do you see the same kind of patterns, for instance, for body mass? Like, is like body mass early in life, like predicting like lifespan, and is it depending on environmental condition?
In berries, you mean?
Yeah.
I don't think we have tested that. I should look into that. I don't know.
Thank you. Very interesting. I wondered if you had any information on how long or if these effects, early life effects, are persisting into adulthood.
We haven't yet, not on lifespan. We know how telomere length reacts with age. So telomere length in our case has this really weird relationship with age. I'll show it to you, which is a threshold model. So we're not really sure if that persists in the long term. So we're now focusing on the early life environment and the direct effects, but it's very interesting to look into that in the long term, but we haven't yet.
It's a very nice talk. I wondered whether you express rainfall in terms of the mean amount of rainfall, but I wonder if what might be more important is the number of days of rain. I mean, I live in Glasgow and it rains every day. So maybe days without rain might be more of a stress than actually going for a long time and then getting a huge amount of rain.
So that might be where the component of variability comes in. So rainfall doesn't follow any clear patterns throughout the year, in Whiteham at least. So we just use the mean and the coefficient of variation in daily rainfall. But I can see how a lack of rainfall might lead to food stress and therefore affect the individual. But something to look into, yes.
Any other questions? No? Oh,
okay. Thank you for an interesting talk. I was wondering if you had looked into time windows on the rainfall and temperature effects.
Yes, I've actually got this prepared. So this is how we calculated the weather conditions. So we have the mean and the variability. And the window, we used a package called ClimWind to optimize that, but it couldn't differentiate an optimal window. So we used the most biologically sensible window, which is the main developmental period. And all cubs are born in February, and the main developmental period is up to May. So February to May is the window which we choose for our weather conditions.
All right, thank you.
okay. Thank you.
Okay, our next speaker is Elizabeth Duxbury, and she's going to focus on fitness consequences of germline mutation accumulation.
Hello, thank you for the introduction. So, aging is commonly considered as the physiological decline of individuals over time, reflected in populations as an increased mortality and decreased reproductive output. Central to evolutionary theory of aging is the concept of the strength of selection on a trait declines with age, resulting in the selection shadow in later life. This led Medawar and later Williams to conclude that deleterious mutations whose effects confined to later life periods can accumulate in populations. Additionally, mutations which result in early life benefits but later life cost can persist in populations as they are invisible to selection, which is weaker in later life. Most prominently, the disposable soma theory of aging proposes that individuals have limited resources and energy to divide between survival, reproduction, growth, and somatic maintenance. Increased investment into somatic maintenance can be at the cost of investment into reproduction, or the reverse, and so this theory proposed the reproductive cost of longevity. There's considerable empirical support for disposable theory, both in empirical studies and also in wild populations. Yet increasingly, we see that lifespan extension is not dependent on the cost of fecundity, and there have now been various empirical studies which show that you can extend lifespan in the absence of a fecundity cost. And so fecundity costs are not an inevitable consequence of increased longevity. And these studies come from work on gene silencing via RNA interference and also from dietary manipulation across many different species. One of the most compelling examples of cost-free lifespan extension comes from the knockdown of the expression of a particular gene in nutrient sensing signaling pathways in C. elegans nematode worms. This gene is DAF2, and using RNAi to knock down its expression, we see that lifespan extension is possible without a cost of fecundity, but timing of knockdown of gene expression is important. So Dillon et al. Showed that if you knock down DAF2 expression from the latest stage of development or in adult life, then you can achieve lifespan extension with no cost of fecundity, as indicated in these graphs. So we propose that this apparent cost-free lifespan extension can be explained by the hidden cost of reduced germline maintenance, leading to a trade-off between parental quality and offspring fitness and quality. Based on the Y model of resource reallocation, the idea is that increased somatic maintenance, but with no effect on fecundity, may have a cost of reduced germline maintenance. As indicated here. So what do we mean by germline maintenance? So this refers to the DNA repair of damage in the genetic material, and also processes such as protein surveillance in the germline tissue. These processes maintain the immortal germline stem cells and the somatic gonads. C. elegans nematodes have a large proportion of their body tissue, which is made up of the germline. Investment into maintenance of the germline is far more costly than maintenance of the soma, and germline maintenance is an energetically expensive process. And so using C. elegans nematodes to study germline maintenance is a powerful system. So previous work in our group has shown that if you reduce insulin signaling in adult parents, then we see increased lifespan in the parents, no cost to their fecundity, and also benefits to the fitness of their offspring, both in terms of Darwinian fitness and also the total reproduction of the offspring. And these results are robust across 3 different genetic backgrounds, as indicated in these graphs. But we asked the question, so in this study, we seem to see that investment by the parents in increased somatic maintenance is accompanied by an increase in fitness of the offspring. But what about the genetic quality of those offspring? And what if we look at the transgenerational consequences over multiple generations? Do we see costs starting to accumulate in the offspring due to reduced investment in germline maintenance by the parents? So to address these questions, I worked on a project to look at does parental lifespan, in the absence of cost to parental fecundity or offspring fitness, lead to increased mutation rates in the offspring and transgenerational fitness costs. To address these questions, I use a powerful study system, C. elegans nematodes, which are useful for aging, evolution, genetics, and transgenerational studies. Because they have a rapid generation time, a relatively short lifespan. There are mutants and powerful genetic tools, such as RNAi interference, to allow age-specific gene silencing. And it's possible to save populations via freezing to do later sequencing work on their genomes. Additionally, the somatic tissue of C. elegans is post-mitotic, so it's no longer dividing, and this makes it more resistant to UV irradiation, in contrast to the germline, which is more sensitive to induced damage, which allows us to experimentally induce damage in the germline via UV irradiation and look at the consequences. So for my project, I use a mutation accumulation approach. So you have a founder population, you take a single individual to establish each mutation accumulation line. That single individual produces offspring from which you choose one offspring randomly to make the next generation. And so across generations, you get successive bottlenecks because you're just using a single worm to propagate the next generation. And so this allows mutations to accumulate in the relative absence of selection. We know in C. elegans, under standard conditions, that the average spontaneous mutation rate in controls is about 1 mutation per line per generation. So using this approach, I conducted 2 mutation accumulation experiments. For the first experiment, and the main point of comparison was, do mutations accumulate faster in sham control lines or in lines where we knock down the expression of DAF2 involved in insulin signaling in adult worms? We allowed mutations to accumulate either spontaneously or we induced mutations using either UV irradiation or a DNA mismatch repair mutant background. And we also conducted the first experiment at 25 degrees, which is a stressful temperature for nematodes, to allow mutations to accumulate faster so that we could see the differences between controls and the DAF2 RNAi treatment faster. For the second mutation accumulation experiment, the design was very similar, and again, we were looking at the difference in extinction between controls and DAF2 RNAi lines after mutation accumulation. But this time we used HERDA1 mutant background. So these mutants lack the transgenerational transfer of RNAi, and so you can look at the effects, purely the parental effects on offspring, without the possible effects of RNAi being transferred and affecting the developing offspring. So what do we find? So from the first experiment, we did indeed, in line with predictions, find faster mutation accumulation line extinction in the DAF2 RNAi lines in blue compared to the sham controls. And this was the case for both when mutations were accumulating spontaneously and also when they were induced either via UV irradiation or in the repair mutants. Then in the second experiment, which I should point out is still running, so these are graphs of the current experimental situation. So again, we found faster line extinction in the DAF2 RNAi lines, in line with predictions, than the sham controls in the irradiated HERDA1 mutants. Which was good because that supported the results from our first experiment in terms of the direction of effect. We're still running the experiment with the control lines, and so it's a bit early to tell, but it could be that effects may differ between the genetic backgrounds. So finally, to conclude, we found from our mutation accumulation experiments that reduced insulin IGF-1 signaling in parents via DAF2 RNAi in adults was associated with faster MA line extinction and faster transgenerational decline in offspring fitness. And this is consistent with reduced germline maintenance as a cost of lifespan extension. We hope to follow this work up with whole genome sequencing of the mutation accumulation lines to directly measure mutation rate between the DAF2 RNAi lines and the controls. And with that, I'd like to thank Alexei Maklikov, all of our group, our collaborators, and you for listening. I welcome any questions. Thank you.
Thank you for this very nice presentation. Any questions? Oh, sorry.
Thank you for your talk, and I got a very quick question. How is the fitness of offspring calculated?
So we look at their reproductive output, so the number of offspring they produce over the course of their lifespan. So we basically, each day, they lay eggs, which then develop into offspring. We count the daily offspring counts. And then we calculate Darwinian fitness, so it involves survival and the reproduction, basically. It's lambda is the measure of fitness.
Any more questions? Over there.
Hi. I wasn't completely sure in your first experiment. Did I understand it right that the RNAi was transferred each generation, so it was knocked down constantly or only in the first generation?
Yeah, so in the first generation, in the first experiment, we weren't using the HER1 mutant. So there's a chance that the RNAi may have been transferred between generations. We don't know that for definite. One of the phenotypic effects of the DAF2 RNAi is interference with development, so you sometimes see developmental abnormalities. We did see that in some individuals, so it's possible they may have received transgenerational transfer of the RNAi, although that could also have been due to other factors like the increased temperature or the fact that they were accumulating mutations. So we can't say for definite, but then using the HER1 mutants in the second experiment, it allowed us to tease those things apart.
Thank you for an interesting talk. There are a couple of studies showing that the lifespan advantage over their controls disappears, of long-lived mutants over their controls disappears when they live in more challenging environmental conditions. And I was wondering if in your experiment the lifespan advantage of the long-lived mutants was decreased or when you expose them to a couple of challenges. I couldn't deduce that directly from the graphs.
So in my experiments, I wasn't directly measuring lifespan, but I guess the time until extinction was some measure of their condition. So in the first experiment, when we used the stressful temperature, they went extinct faster than they are so far in this more benign conditions. So I guess that's one indication. Yeah, stress could be different. Thank you.
Okay, the last speaker of this session is Tom Torgenza, and he's going to talk to us about senescence in wild crickets.
Okay, thank you very much, and thank you very much everyone for hanging in there. Second day of the conference is always the hardest. The work I'm going to talk to you about has been done out of our University of Exeter's Penryn campus in the far southwest of the UK, and it's all been funded by NERC, for which we're very grateful. And the vast majority of the work's been done by my colleague Rolando Rodriguez Munoz, but recently we've been doing a lot of work with Jelle Bornekamp and Paul Hopwood. So the thing that inspired us to start looking at aging was really the fact that there'd been a really large increase in the number of studies of aging in wild populations over the last 20 years or so, but there remains an extreme shortage of studies on invertebrates, which is unfortunate because obviously the invertebrates are quite numerous, and a lot of laboratory studies of aging have been done on invertebrates. So we thought it'd be interesting to see what's really going on in relation to senescence in the wild. So the questions that I'm going to focus on today are: do annual insects decline in survival probability and in their phenotypic performance with age? Does actuarial senescence reflect phenotypic senescence? And are patterns of senescence sensitive to natural variation in environmental conditions? So I'll try to cover those questions. So the model system that we've been using to try to study this, well, I say model system, it's basically just a field with a load of crickets in it, is this typical annual insect, the field cricket. It has a single generation every year, overwinters as a late instar nymph, and then in the spring, the individuals start becoming adult, and then they live for only a few weeks. And the handy thing is that they're obligatorily associated with burrows. Both sexes build burrows, so we can find them quite easily. So this is a shot of our meadow from above, and this is like all the burrows that there were in that meadow in 2016, I think. So they're confined to this neat little area here. And when we land the helicopter, we get a view like this of the meadow. So it's a typical temperate meadow that you find throughout northern Europe. And what you find if you walk very quietly around the meadow and hang around is these burrows with crickets living in them. And what we do in the spring is we go out into that, collect all the crickets one at a time. This is my colleague Rolando, who's been abusing crickets for so long that he's now violently allergic to them, which is presumably some sort of karmic rebalancing going on. But we can tag, he's here sticking a tiny tag onto the cricket. We stick a unique number onto every cricket, put them back into their burrows, which they seem to be fairly unfazed about. We then have a network of around about 140 digital video cameras with infrared on them, recording them all day and all night, which sounds great, but of course then we have to spend all the rest of the year watching close to 1 million hours of video that we've now watched. We now have a large database about all the detailed behaviours of these crickets and their life history traits. There's around about 11 years of monitoring more than 2,000 crickets and vast numbers of behavioural and life history events. So we've been spending so much time collecting data, we haven't done lots of interesting analyses. So if you've got questions you'd like to point at this data set, then come and speak to me. But an obvious thing to start looking at was aging, because we have really detailed information about exactly when the crickets become adult, when they're born. So this is a cricket emerging as an adult. We know the precise time that happens. We also know very accurately when the nastier end of their lifespan happens. So kind of horrific attacks like this are common. But I can't watch anymore. We also see just crickets just wandering down their burrows and never coming up again. So this gives us the opportunity to look at actuarial senescence, to look at the demographic population level measures of senescence. We can observe adult emergences and deaths, and we can fit a mortality function like we've heard lots of speakers talking about today. We find that a Gompertz distribution fits the actuarial data very well. And we can also measure phenotypic senescence because we can see everything they've done through their lives. So the thing I'm going to focus on today is the singing that males do. And this is a male hanging out next to his burrow. So we can check every hour on whether they're singing or not, or we could check all the time if we wanted to. We also see other things. This is a male being displaced from his burrow by another male, but we get detailed information about how much males sing and how that changes through their lives. So we can ask the question, do the patterns of actuarial senescence that we see in this population match the patterns of phenotypic senescence that we might see? And this is kind of a thing that we all assume, that aging in populations, increased probability of death with age, is going on because individuals are aging, but we rarely actually have information showing that in years where individuals are aging faster, we're also seeing more actuarial senescence. And that's because you need lots of independent generations to look at that. And often with long-lived vertebrates, you haven't got enough independent generations. So if we look at 10 years of this cricket study, this is the two parameters from the Gompertz distribution. So if we just concentrate on this B1 parameter, this is the slope of the relationship between age and mortality. So this is how much more likely you are to die as you get older. So if that parameter is greater than 0 here, it means that there is actuarial senescence. And you can see that in nearly every year, there's quite strong evidence of actuarial senescence. As males get older, they become increasingly likely to die every day of their age, that they age. The pattern of phenotypic senescence is a little bit more messy, a lot more messy really. But if we just take an example, this is 9 years where we've actually managed to get all the video watched and so on. And this is all the male crickets in, say, 2016. You can see here that once males become adult, they increase in the amount of their singing and then they decline. And these are days on the bottom here. And we've done the analysis using within individual corrections for selective disappearance and so on. But there's clear evidence that in general, there's a decline in singing activity with age, but in some years it's much more obvious, and in other years it's more or less absent. So that gives us the opportunity to ask that question, do the years where phenotypic senescence is more obvious, are they also the years where we see more actuarial senescence? And the answer is that they are. So we look at this graph here, this is, for instance, this is a year where actuarial senescence was very strong. There's an increased likelihood of mortality with age. And it was also a year where the decline in singing was the most pronounced. And if we just do a simple correlation there, there's quite a persuasive relationship between those things, which is reassuring because this is kind of what we all thought was going on. The second thing we could kind of have a look at was the prediction from life history theories of aging that the environment should impact upon senescence. And so an obvious environmental parameter that might be expected to affect senescence in males would be something like how much they had to compete with other males. And so demographic factors are likely to be important. And for reasons that we don't understand very well, in fact we don't understand them at all, there are substantial differences in the sex ratio among years. So differences in the sex ratio of newly emerging adults among years. So these pink years are 3 years where the population was very female biased, nearly twice as many females as males in 1 year at the point when individuals became adult, whereas in these 3 years there's more or less even sex ratio. So we can compare those years. And if we look at actuarial senescence in those years, you can see the B0 parameter, just the baseline mortality, is exactly the same between those years. But if we look at the rate of increase in mortality with age, the B1 parameter, there's a significant increase in that parameter in the years where there are more males. So another way to look at that, these blue years are years where there were a similar number of males to females. In these pink years, males were having it easy because there was an excess of females in those years. And males are aging faster when they're having to work harder because there are more males around. We can again look at the phenotypic evidence in relation to that, and this is even more messy. It's the difficulty of looking at insects in the wild. Everything's very messy. But if we just look at the strong female bias years, this is the years when males ought to be having it easy. There's really no evidence that this is the within individual change in calling activity. As individuals get older, there's not really any evidence of any change. But if we look at the blue years, then as individuals get older, they decline in the singing activity. So whether or not males decline in calling depends on how many females are available. And if we do the analysis properly, this is quite a significant effect. So just to summarize all that then, both actuarial and phenotypic senescence are occurring in these insects that are only living for a few weeks, but the patterns vary quite a bit between years, both actuarial and phenotypic senescence. There's a strong correlation between those things, as we'd expect. And also senescence is environmentally dependent. So in years where males are more common, they senesce faster in both measures. Okay, thank you very much.
Okay. Thank you for this very nice presentation. Any questions? Yes?
Thanks for that. The last thing about the more males leading to faster male senescence, because your data is so amazingly individual based, can you test if it's that those particular males who have been involved in a lot of fights, that kind of like wears them out?
Yeah, we can look at that, and we have looked at that, and there wasn't a clear pattern in relation to the number of fights they have. But I suspect that is a power issue. So even with 1 million hours of video, the number of fights males have would be sort of between 5 and 20 in a lifetime or something. So we didn't detect that effect, but it's not definitive evidence that that's not the case.
Any other questions?
Thank you. You got really a very cool system. In your first slide, there seems to be a temporal association with. It seems that actuarial senescence increases through time. I was wondering what was going on in your population, if there was something going on in your population.
Okay. So you're saying when we looked at all those that you spotted, that there it looks like it's increasing through time?
Yeah.
I would be reticent to do that. I think it's dangerous to look at these kinds of things and start spotting patterns. Our human brains are evolved to look for patterns like that. If I did that, I would be seeing patterns everywhere. So there might be, but I think we need to run it for another 90 years or something to really be confident about that. Okay, thank you very much.
Okay, thank you very much.
To finish, I just would like to thank all the speakers and participants of this symposium who gave very nice presentation and very nice posters. So please. And just a quick reminder for the people attending the symposium dinner. So it's at 6:30 at Sergio's. It's the place along the river, and so you can either meet us there, or we're going to leave from outside at